All right, so let's talk a little bit about the process of skeletal muscle contraction and relaxation. Carry that away. So all skeletal muscles are innervated, excuse me, connected to a neuron. A single motor neuron communicates with many muscle fibers. Each connection is called a synapse. The neuromuscular junction is the synapse where a single motor neuron communicates with many muscle fibers, transmits its signal, the nerve impulse, action potential, from the neuron to the sarcolemma of the muscle fiber. Three components of the neuromuscular junction. You have the axon terminal, which contains the synaptic vesicles, which are filled with neurotransmitter acetylcholine. Neurotransmitters are chemical triggers changes in the target tissue, allows the cells to communicate, and it goes back to that core principle. The synaptic cleft is the space between the axon terminals and the muscle fibers. It's filled with collagen fibers and a gel that anchors the neuron in place. And the final component is the motor end plate, a specialized region of muscle fiber plasma membrane, a special region of the muscle fibers plasma membrane. Has a folded surface, contains many ligand-gated sodium channels. The acetylcholine is ligand that opens the gates, allows the sodium to diffuse into the muscle cell. So there's the motor neuron, there's the axon terminals connected to the synaptic cleft. There are the acetylcholine receptors. There's the acetylcholine in little bundles. There's the voltage-gated calcium channels, which allows the calcium to come in. And again, neuromuscular junction video that you can watch at your leisure, and an additional one as well. Skeletal muscle contraction. Muscle contraction phases, you have the excitation phase, the excitation contraction coupling, and then the contraction phase. During the excitation phase, action potential from the brain of the spinal cord arrives at the synaptic terminus of a motor neuron. Acetylcholine is released into the synaptic cleft. The acetylcholine diffuses across the cleft, binds to the ligand-gated channels of the motor end plate. The ligand-gated channels open, which allows the sodium ions to enter the muscle fiber, generating end plate potential. The end plate potential, which is a local depolarization, resulting of the sodium ion influx. In other words, the sodium ions coming into that area. Multiple end plate potentials typically require to stimulate action potentials in the surrounding membrane to trigger functional muscle contractions. The motor neurons continue to fire action potentials. Acetylcholine is rapidly degraded by acetylcholinesterase in the synaptic cleft. There's the synaptic cleft. There's the axon terminal. There's the sodium. There's the motor end plate. There's the sodium binding or sodium channels. There's the acetylcholine. Calcium comes in. That's what triggers the release of the acetylcholine. The acetylcholine binds, opening up the sodium channels, allowing the sodium to move in and create the action potential. Another video for you to watch at your leisure. Couple of review questions. So the end plate potential is generated by the influx of what into the motor end plate? Sodium. Acetylcholine is released from the synaptic terminus in response to, a couple of minutes to think about that one, an action potential arriving at the synaptic terminus. The term synaptic cleft refers to fold in the motor end plate, a vesicle in the synaptic terminus, gap between the neuron and the muscle fiber, or the space between adjacent muscles. The gap between neuron and muscle fibers. Sodium channels on the motor end plate are ligand-gated channels. In-plate potential is it an action, a local repolarization, a local depolarization, or a local hyperpolarization? A local depolarization. All right, skeletal muscle contractions. Excitation, contracted coupling are links between events at the neuromuscular junction and the contraction. In-plate potential opens a voltage-gated sodium channel in the sarcolemma surrounding the motor in-plate, triggers an action potential. That action potential propagates or moves along the muscle fiber. Depolarization of one area of the membrane triggers the next few voltage-gated sodium channels to open. Process continues in a chain reaction to the triad. The action potential signals terminal cisternae to open the voltage-gated calcium channels, which releases calcium, again, 2, meaning they've got 2 extra protons into the cytosol. So here's the action potential moving down. Once the signal gets down here, sodium channels open, allowing the sodium to move into this system in the triad. Another video that you can watch at your leisure. The channels that open the sarcolemma surrounding the motor end plate and generate action potentials are the theme of Jeopardy plays and voltage-gated channels. Propagate refers to an action potential, means does it stimulate it, does it inhibit it, does it magnify it, or does it spread it? It spreads it. Remember, propagate means to disperse. In order to trigger a muscle contraction, an action potential must reach the triads. A triad consists of 2 terminal cisternae and a T-tubule, an M-line, 2 tubules and a terminal cisternae, 2 terminal cisternae and a T-tubule. What is released from the sarcoplasmic reticulum in response to the arrival of an action potential? Calcium. All right. Muscle contraction part 2, another video showing the animation of the videos, or the animation of the process. In preparation for muscle contraction, calcium ions are released at the terminal cisternae, bind to troponin. Troponin has 3 subunits. One subunit binds calcium, one binds actin, the other one binds tropomyosin. Tropomyosin moves. The active sites of the actin are exposed. Again, here's where that myosin head comes up here and attaches on the actin sites in response to calcium being dumped in. There's a nice little video that shows that. Tropomyosin, does it cover actin active sites? Does it bind with calcium? Is it a small globular protein, or does it have 3 subunits? Covers actin active sites. Troponin has 3 subunits. Which of the following does not bind to one of those subunits? You just went over this. Myosin. The actin, the calcium, and the tropomyosin all bind to troponin. Choose the correct answer, the sequence of events that occur in preparation. So you've got A, B, C, and D. They're all parts of the system. What is the order they should be? So the action potential arrives at the triad. Calcium is released from the terminal cisternae. Calcium binds to troponin. Tropomyosin exposes the active site. Tropomyosin exposes the actin active sites. Calcium binds to troponin. The action potential arrives at the triad. Calcium is released from the terminal cisternae. Calcium is released from the terminal cisternae. Calcium binds to troponin. Active potential arrives at the triad. Troponin exposes the active site. So which one of those 4 is the correct sequence of events. Give you a moment to look at them. I've chosen my answer. Let me see how we do. Hey, I was right. And the only reason I said that is because the action potential arrives at the triad was the first thing that I was 100% certain that that was the first thing that happened. So, again, that's one of those ways of answering these questions. I knew that this was the first thing that happened, so it could be the only one that was right. Contraction begins when the actin's active site is exposed, initiating the cross-bridge cycle. The myosin head cocks once. ATP is bound and the energy is gathered by hydrolysis. In the high energy position, with the third phosphate still attached, the head is able to bind to an active site of the actin. The cross-bridge is at a 90-degree angle relative to the thick filament. Power stroke, as this extra phosphate is released from the head, the myosin pulls the actin toward the M line as it pivots to its relaxed or low energy position. The cross bridge is at 45-degree angle relative to the thick filament. So basically it's moved from here to here, from a 90 to a 45. Myosin can then bind to another ATP, breaks the link with the active site. Detachment does not allow the thin filaments to slide backward because some myosin heads are still attached. And again, here it comes up. See, it's at the 45-degree angle. Moves up to the 90, stays at the 90, drops back to the 45. But what it has done is it has pushed this whole filament that way. And since there's another one over here, and another one over here, and another one over here, still bound to it, it doesn't, just in case you couldn't see my pointer, there are more of these off camera, and there are more of these off camera. So when this releases and goes back to this lower position, some of these other ones are still holding the attachment. That doesn't let this filament slide backwards. It holds it. So this moves it, and then it slides and holds. It is not until all of these release that you can then see that the fibers will slide back across each other the other direction. Again, this is a nice little video. I've watched this one several times to show you how that sliding function works. Hydrolysis of ATP is responsible for which of those things? Recocking of the myosin head, taking it from that 90, from the 90 to the 45-degree angle. The binding of ATP to myosin is responsible for release of the myosin heads from the active sites. The release of ATP and the phosphorus ion from myosin occurs during the power stroke. That's when the filaments slide across each other. The myosin head returns to their lower energy relaxed state during the power stroke. Power stroke pulls the thick filaments toward the Z lines, positions the myosin head in their high energy position, shortens the length of the filaments, or pulls the thin filaments toward the M lines. Remember, we can throw this question out because one of the first things we noted was that the length of the filaments didn't change. Contraction cycle may repeat as long as the stimulus to contract continues and the ATP is available. The myosin head recocks, binds to the first actin molecule, and the power stroke repeats. The myosin binds to a second actin, and so on, over and over and over. For the average contraction, this process will be repeated from 20 to 40 times for each myosin head in each sarcomere. So again, there is this power stroke bringing these things closer together with There it's attaching to actin number 1, then it's actin number 2, then it will connect to actin number 3, sliding this thin filament over the thick filament. Another video. Linking cross-bridge cycle to its sliding filament mechanism. Sailors pulling on an anchor rope. The rope represents the thin filament. The anchor is the Z-disc. The hands of the sailors are the myosin heads. The hands of some sailors are holding the rope, while the hands of other sailors are pulling the rope. Some are repositioning their hands farther up the rope, allowing their arms to rest. They work together to pull the rope without allowing it to slide backwards. It's also true of myosin heads. Some pull the thin filaments toward the M line, others detach and are reactivated by ATP. Filament slides in only one direction toward the M line. Skeletal muscle relaxation. I'm going to stop there for the today, and I will pick back up a little bit later on Wednesday, and we will talk about skeletal muscle relaxation.